Okay, microbiology friends. In this video, I'm going to be drawing the pentose phosphate pathway, also known as the pentose phosphate shunt. And a shunt is a shortcut or a detour or a side quest. It takes us away from some other path or pathway, as the case may be. So we're going to start with glucose 6-phosphate. And I think you know how we get that. You should know by this point how we get that. We start with glucose, and then hexokinase can consume an ATP to phosphorylate glucose 6-phosphate. And from there, we can convert that into 6-phosphoglucono delta lactone. And that seems like a mouthful. We're going to use an enzyme called G6P dehydrogenase. And when you see a dehydrogenase, get ready to be harvesting some electrons. And in this case, we're not merely harvesting NAD, but the phosphorylated version, NADP plus, and that is going to give us what it usually does, which is NADPH plus a proton. And then that intermediate is going to get converted into 6-phosphogluconate and 6-phosphonogluconate is going to be in turn converted into ribulose 5-phosphate. And while these two intermediates might sound like some kind of gibberish, ribulose 5-phosphate might sound just slightly kind of sort of a little bit familiar-ish to you. And how do we get there? Well, we're going to use 6-phosphogluconate dehydrogenase. So another dehydrogenase. So again, we're going to get another molecule of NADPH plus a proton. And we're also going to release a molecule of carbon dioxide. So we start with a 6-carbon sugar, convert it into a couple of other 6-carbon intermediates, And then we end up with a 5-carbon sugar, which is ribulose, and that would be our pentose. That would be our pentose. Okay, at this point, we are done with the oxidative part of the pentose phosphate pathway. In those reactions, because we are harvesting those high energy electrons, they are irreversible. And we're pivoting to the second part of the pentose phosphate pathway, which is the non-oxidative part. And these reactions are reversible. Isn't that exciting? Well, what's that going to give us? Here we can take that ribulose 5-phosphate and we can convert it into a number of different things. So here, reversibly, I'm going to go this way, and I'm going to use ribulose 5P isomerase. And we're going to isomerize our ribulose 5-phosphate. And that is going to give us ribose 5-phosphate. Now that should definitely be ringing some bells, because where do we find ribose? In ribonucleic acids, you betcha. So ribose 5-phosphate, we've got the ribose, we've got the phosphate, all we need is a nitrogenous base, and we've got some nucleotides. Well, really, they're going to be they're not deoxy just yet. That step's not so hard. But these are the ribose 5-phosphate is going to go into nucleotide synthesis. But another thing we can do with the pentose phosphate pathway is this is going to be ribulose 5-phosphate epimerase. And that is going to give us xylulose 5-phosphate. Which we can take these two molecules. Again, everything here is reversible. Run them through an enzyme called transketolase. And that's going to give us a molecule of G3P and a molecule of cetoheptulose 7-phosphate. So we've got a 3-carbon phosphorylated sugar and a 7-carbon phosphorylated sugar. And we can take those, run them through another enzyme. So instead of transketolase, now we're going to use transaldolase. And that's going to give us fructose 6-phosphate and erythrose 4-phosphate. So 10 carbons here. Racemophu through transketolase, we get 10 carbons here, only now instead of being 5 and 5, it's 3 and 7. Racemophu, once again, we get F6P, fructose 6-phosphate, and erythrose 4P. And we know that F6P can get fed back into Embden-Meyerhof-Parnas, or regular old glycolysis. Erythrose 4P can be combined with xylulose 5P. So now we've got 5 carbons, and we can put that through those two things through transketolase. Same enzyme, and we're going to get, so we've got 9 carbons here. We can get a molecule of F6P and a molecule of G3P. And both of those can make their way back into Embden-Meyerhof. Just going to make a shortcut there. So this pathway is basically doing a lot of molecular horse trading in terms of where the carbons are going. So we go anywhere from a 3-carbon sugar up to a 7-carbon sugar, with 4-carbon, 5-carbon, 6-carbon sugars all in the mix as well. So you might be wondering, what do we get out of this particular reaction? Well, for one thing, we get 2 molecules of NADPH from the oxidative part of the pentose phosphate pathway. So this does a couple of things. One, it is reducing power for anabolism. So we can build up more complex molecules from simpler molecules. We can make polymers. The other thing is reducing power for antioxidant activity. Which means we can use those electrons to help get rid of reactive oxygen species. ROSs, like hydrogen peroxide, superoxide. So that's the benefit of having NADPH, and the benefit of having all of this sort of molecular shuffling down here, producing all these sugars that have different numbers of carbons in them, is that these can also be used for different feed into other pathways to make other biological molecules. Just for example, nucleotides, like we see here, having ribose 5-phosphate, ribulose 5-phosphate, that can be fed into photosynthesis because the carbon dioxide acceptor in the Calvin cycle is ribulose 1,5-bisphosphate. So if we phosphorylate ribulose the second time on the other end, we can do the Calvin cycle. But who is actually doing this? Many species can do the pentose phosphate shunt, but oddly enough, it is absent in Archaea. So Archaea, not able to do this pathway. So overall, what's happening in this pathway is that we are going to oxidize glucose 6-phosphate, I'm going to abbreviate it as G6P, to ribulose 5-phosphate and convert it to a variety of, well, I'll just say 3 to 7 carbon sugars, which can be used for as feedstocks into other biological molecules. So how do we make things like amino acids, vitamins, things like that? So we can use these molecules because these reactions exist. We can make other things from these different molecules. So it's biosynthetic. And that is your pentose phosphate pathway.